Afternoon, and welcome to my talk. I am Nathan Brouwer, and I'm going to be talking about how we can use Python and tools in data science and machine learning to analyze genomic data, including data that possibly you've had collected on yourself through 23andMe. And I'm running a little poll using Slido here, and let me see how the results are going. It's totally anonymous, and it looks like most people at least know people who have had genomic testing using a service like 23andMe, and a lot of you are interested or would be open to that possibility. So cool. Hopefully you can learn something about genomics here and how you could use this data or go on and get open source data and play around with it. Also, I should say I am not with 23andMe. I'm just co-opting the name. There's many companies that do this sort of thing. They just have the catchiest name. So I am a biologist and a data scientist. I spend all my time thinking about organisms and cells and DNA, and I use tools in Python like scikit-learn in order to study it. And I'm going to talk about today one of the intersections of biology and machine learning and data science, which has the fancy title computational genomics, which includes a lot of different things. But if you spit in a tube and send it to 23andMe or sampled for some other reason, my dad has cancer and he's had genomic screening done for the cancer type he has. If you do that type of testing, you are participating in a computational genomics workflow. So that's what I'll be talking about today. Before then, though, I want to take a step back, talk about just about data science. And this might be a fringe opinion, but I really think that we are in a crisis in data science and machine learning, and it's a crisis of boring data. Does anyone have a machine learning or deep learning book on your bookshelf? I bet every single one of those books uses one of these or both of these data sets, the iris data set and the handwriting data set. And there's reasons to use these data sets, but at least the iris data set's been around for over 100 years. It's getting a little old. And I'm a big believer in using data that's interesting and exciting. So genomics data is cutting edge. You can spit in a tube and create more data by the end of the week. It tells us how the world works. We actually learn something by looking at that data, and we expand our knowledge of the world, and we can learn about ourselves. We can actually learn about our own ancestry. So for students and people learning, there's a lot of investment or potential investment in this. Also, it has tremendous societal relevance. There's ethics about data storage and the type of data storage and things we can learn about ancestry with them. So there's all sorts of cool aspects of this. However, genomics data is complicated, and it's easy to misinterpret, and it has tremendous societal relevance. So misinterpreting it can have some downsides. So let me turn this on, but I'm really interested. I'm kind of workshopping these ideas, and this is the first time I've used Slido. So hopefully this will work. I think I just switched to a new open text poll. But if you are interested and you have any questions, I can try to get to them at the end, or I'd be happy to talk. Please, any questions, concerns, things that look weird, things that look exciting, please give me some feedback on that, because I'm really, really interested in how this goes down. And in case anybody didn't get it, the next slide, though, is a slide from the program handout. I'm really, really interested in how we can expand the type of data we use in teaching and learning and blog posts and that sort of thing. So there's been some other talks on neuroimaging and data on chemical analysis that has relevance to the opioid crisis. So there's been other talks that have kind of been on this theme on other data we can use when we're practicing our programming and teaching about data science and that sort of thing. So there's some sections to my talk. First, we're going to do a little crash course in genomics. I'm going to call that Hello Genome. The next part is actually what 23andMe gets to you when you send it as a sample. This is all your base now belong to us. And then here's just a gratuitous puppy picture that we'll play in later. And then at the end, we're going to send in the droids, and we're going to talk about how we can use these data with machine learning and deep learning. So Hello Genome. So really basic biology, just to make sure we all have a starting point. So what is a genome? And that's just the entire set of DNA sequence in something. So if you've ever seen a picture like this in a textbook or online or a magazine, that's a kind of mile-high view on a genome, all of the data that's in somebody's cells. And so if you're looking at a chromosome, you're looking at a genome or part of a genome. And as you're perhaps remember from biology class, the sequence of a genome is literally, we can represent it with the letters A, C, G, and T, and that's what it boils down to. So all of this stuff here, we can characterize all of the information in this just as a sequence of A, C, T's, and G's. There's a little bit more to that, but you can take a biology class for that. So before I go on, I want to step back and just appreciate the scale of data. Like genomic data is like one of the original sources of big data. I'm going to show a plot here. The x-axis is going to be chromosome length. Note that the scale is 1 times 10 to the 8th, and then the y-axis is going to show you different chromosomes. And the smallest chromosome in human cells has 50 million letters in it, 50 million A, T, C's, and G's down here. The smallest ones, 20, 21, 22, 23. And the largest ones have 250 million letters in them. So very, very large amount of data gets generated when we do this. By way of comparison, if you have slogged your whole way through all of the Wheel of Time series, that's maybe 3 million letters by my estimate. So that puts you at the scale of like one chromosome, the smallest chromosome. Once we start looking at DNA, we can look at one person's DNA and learn a lot about them, but a lot of research involves looking at multiple people's DNA sequences, how they compare and contrast. So I might have a G at a particular position and say the sequence is A, C, G, T, and someone else might have A, C, C, T. And that variation, when we look at it between people or across lots of people, can tell us a lot of interesting stuff. And I just put that in this morning, and I forgot that it was coming up, but I have to talk about it. We have to give a shout out to Loki when we talk about variants. We call these different locations variants. They have lots of different names. I will try to call them variants today, but there's way too much lingo in biology as it is, and then we come up with multiple names for things. So that's one of the problems of genomics, is having a new lingo and a diverse lingo for things. Now, DNA, as I already said, as you probably remember, has 4 letters, A, T, C, and G. But luckily, while there is possible 4 different versions of a variant that can be at any different place in the genome, so I could have G and you could have C and someone else has A and T, luckily it's usually much more simpler than that for various reasons. I might have a C and you might have a G, and everyone else has C's in it. So it's much simpler. Usually we're just dealing with a binary situation, one version or the other. So we just get Loki and Sylvie. Okay, so that's our crash course in genomics. Now we're going to say, like, when we give our genomic sample, when we give a tissue or a blood or some other sample, a spit sample, what kind of information are we giving to a lab or to a corporation? And so only a few people seem to have ever actually used a service like 23andMe, but lots of other folks are interested, so this is what the kit will look like when it shows up in the mail. And there's lots of actually other companies that will do this. There's Ancestry and a bunch of other ones that I didn't realize. And these are all companies that provide some sort of kit that you provide a DNA sample, they extract the DNA, they do the fancy computer stuff, and they're able to characterize your ancestry and come up with some descriptions also related to health. Key thing here is I say ancestry characterization. Sometimes these are called ancestry tests, like there's a right or wrong answer or a true and false answer. Ancestry is a very complicated thing, and so characterizing, estimating, approximating is a much more appropriate term. And estimates are about 25 million people, or approaching 10% of the US population has one of these kits done. I think it's especially interested, people in the United States are especially interested in this. And I'm focusing just on 23andMe because it has a catchy name and it's one of the more popular ones, but I'm not affiliated with them. A little aside, I wanted to get an ancestry test this summer so I could have my own data to plug into this. and the rest of the family wanted the puppy. And so this is Theoden albus. We've had him for a week. He's great, but I still don't have my ancestry test, so maybe next year. And if you don't have your own data, or it's kind of like talking to ChatGPT. You are giving it information by interacting with it. You are paying to give something to a genomic testing company, and you're getting back something, but they are keeping that data and adding it to their database and continuing to analyze it. So if you don't have your own data or aren't comfortable providing it to a for-profit company, luckily there are places you can get other people who have decided to post their data online. So you can go to the personal Genome Project and download data from people. So thank you to the people who have participated in studies and who have posted their data for free. Also, let's talk at the end, there's ways that you can just simulate data. And I happen to use a data set that was posted by somebody else, Marielle Vols. I don't know if it's her own data, but in my journey, I happened upon it. So thank you to them for posting some code, and that was useful, in particular posting some data that I found easily. Now, if you submit a sample to an ancestry company like 23andMe, you can get a big fancy report back that tells you about ancestry and your DNA and some health stuff. But what I want to talk about today is what happens when we download the raw data to that and what we can do with it ourselves, and then how actually they are getting some of this fancy information from that raw data. And so this is what it looks like. It extends another 985,000 rows. Below that, it's very, very large data. And this is something you can easily load in if you use pandas. In Python, it goes in pretty easily, and this data can start to get very big. So the new package is polars that I found very useful as I start to work with larger and larger data sets on my somewhat sad old tablet here. And I'm still getting the code, thanks to Theo. I'm still getting the code for this talk online. There's just a stub there on Colab, but I'm going to start providing code to replicate all of this stuff. So again, here's the text file, and this top part is just metadata. I'll provide some notes in my code for some of the stuff that's interesting up there. So this is kind of the meat and potatoes of what we have. It's just 1, 2, 3, 4 columns, but then it extends down to a total of about 900,000 rows of data. And each row, in this case, is a place where humans can vary and gives you what genetic material you got from your parents. So here is a single position in this person's genome. And we would call this a feature, or we can turn this into a feature. We can turn this into information for machine learning. Each of these variants has to occur somewhere in a genome on a chromosome. So these chromosomes are listed there. And RSID is a ID number, a ID code used by Genomics, and it's somewhat stable and somewhat commonly used, but one of the problems with genomics data is it's a rapidly evolving field, and for various reasons, it's hard to pin down exactly where things are. So things like this are always changing, but it is a useful number. You can actually take these numbers, Google them, and see if they pop up and find out information about those particular variants. And then genotype, this is the actual data. This tells you what genetic material you got from each of your parents, and then this is what we will turn into features and use in our machine learning. And some basic biology review again. CC, we call this homozygous in biology. This means you got a C from your mom and a C from your dad, and you are CC. AG means you got an A from one parent and a G from another. And I'm not going to go into it, but you have to do a few little things to convert this into numeric values in order to do the math on it with machine learning. And just with that data, there's some fun stuff like I mentioned, like you can look up the trait that whether you can smell asparagus in urine. There is a genetic variant, has absolutely no evolutionary purpose as far as I know, but some people can smell asparagus and other people can't when people go to the bathroom. Very bizarre, pointless trait, but you can look that up in a set of genetic data. This person couldn't, I don't think. Again, let's step back. I always want to emphasize the immensity of what we're dealing with here with the human genome. This data set here has over 900,000 rows of data, over 900 variants in this data set. But our smallest chromosome is on the scale of tens of millions, and it goes on up to hundreds of millions. So when you get 23andMe to characterize your genome, we're looking at a tiny, tiny fraction. Actually, I did plot it over there. It's 0.3%, but it's just a little tiny, tiny sliver. That's actually the amount of information that is being used in all of that. And then there's the rest of the genome. And actually, if you go to public data sources that are available and get kind of the most thorough, up-to-date surveys of diversity in the human genome, you can max out at about 4% over there in a project called the 1,000 Genomes Project. And then there's the rest of the genome. So we're dealing with very vast amount of data, 900,000 rows. It's pretty big for biology, but it's a drop in the bucket when it comes to the human genome. So why is it that we're using a lot of data, but it's only a small fraction of the human genome when it comes down to it? Why is that? And that's because all of this orange, humans are basically identical. There's some caveats and asterisks there that you could talk to me about. But basically, for most of our genome, we're all identical. And the other 4% is places where some people vary from each other, but many, many people are identical in those sections. So the current statistic is about 98, most humans are about 98% similar if you compare their sequences to each other. So all we're doing when we do genomics is we're ignoring all the stuff that makes us identical, and we're zooming in on the places where there's potentially variability. And there's actually, because we have 900,000 pieces of information, we can actually extract a lot of data. All right, so now we've arrived to the machine learning part. So we're going to talk about how do we take our data, how do we also take some public data and do some deep learning and some machine learning and some data science. We're going to send in the droids. And when you are working with genomic data, again, comparison is really, really important. There's some interesting things you can do if you just have your own file and your own data file, and I'll post some code for that. But things get really interesting when you start looking at lots of different genomes and then comparing yourself to that. And the 1,000 Genomes Project is a big open access database of anonymous data, and it's still kind of clunky to use, but it's the easiest one out there, and I've used it a lot in teaching classes. And I'll call it the 1KGP for short. And it has over 2,500 people from 25 different populations from around the world who volunteered for this study and submitted DNA samples. And the data is all publicly available and anonymous. So it covers all of the continents and lots of different places from around the world. And this is probably the most thorough survey of genetic variation in humans, and they identified about 125 million different variants, but we have a genome that's in the billions, so it's only a portion of all of the variation. And I'm not going to directly use 1,000 genomes. I'm actually going to be getting data from this paper. They synthesized some 1,000 genomes data and added some of their own. And this is a really, really handy data set because they just posted an Excel file online with all of the data. It makes it really easy to kind of grab and go and do some work with. And these folks, Shing et al., you can tell I'm an academic because I have to put the citation in there. They looked at fewer than 1,000 genomes. They looked at just 250,000 genetic variants from just 850 people. But they looked at 40 different locations around the globe, 40 different populations throughout Africa, Europe, Asia, and a few in the Americas. And at the time especially, this was a ton of data. Things have gotten bigger since then. And it adds up to a 615 megabyte spreadsheet when you download it. So that's where Python can chew through this pretty fast. But some of the data cleaning steps, since I don't write that efficient of code, take a little bit of time. But I'm working on that. However, this is a great data set. It's fabulous to use. It's pretty similar to what 23andMe is actually using, or it has a lot of important analogs to it, but it's still most of the world's genetic diversity is not captured. And you can look at like, oh, there's lots of dots, that's great. But when you kind of block out the dots and look that everything is left, there's no information about the peoples who live in all of these different places in this data set. And really 23andMe is bigger and better, but not like an order of magnitude bigger and better. So anytime you look at genomics data, you should remember this is what's not being represented. They're kind of smoothing over these gaps. A lot we can learn, but still a lot more data to be collected. Luckily, there is a 1,000 African Genomes Project that is going forward. China has its own genomics initiative, so there's making a lot of progress in better understanding genomes, but not all that data is publicly available. A key missing piece in terms of data and also biology is that there's no information here from North Africa, the Mediterranean, Saudi Arabia, the Levant around Israel and Palestine and Syria and Turkey and all of that. So this kind of interface between African populations and the rest of the world where humans migrated through on our way to colonizing the world, that is absent from this data set and most data sets, including 23andMe. So this is a very important missing piece, and it influences the types of results we get when we toss this into machine learning. So machine learning is a huge, vast field. This is kind of just meant to give you a snapshot. If you don't know what any of this is, don't worry. But one of the cool things about genomics data is it's kind of a one-stop shop for interesting things you can analyze. You can almost any machine learning or deep learning algorithm can be used for genomic data. So for teaching and learning, you can take genomics data and then run with it and use that for lots of interesting things. I'm going to start off with a technique called PCA. Mathematically, it's pretty complicated. I'm just going to give you a little bit of description there, and you can go watch StatQuest videos and other things that give great descriptions of what PCA is. And we can do all of this in scikit-learn very quickly in Python. So the top of my data set, once I've done all my processing, and I'll give you the scripts and you can do it yourself, the top of my data set is going to look like this. Each row now is a person, an anonymous individual. And there's 800 or so more of these that Xing had in their data set. And then a key thing is that each column is a variant. We would call that a feature in machine learning. And after all my data processing and cleaning is done, there's still another 15,000 rows, or excuse me, 15,000 columns extending that way. So it's still a very large data set. And when you get a data set, you might have heard this advice before in statistics or machine learning, but you should plot your data. You should get out your artistic impulses and make some graphs. But how do you do that when you have 15,000 columns of data, let alone when if you were actually doing this professionally, you'd have like 785,000 columns of data. So we can try to toss this into MATLAB, and Matplotlib. And a key common tool when you're exploring data and statistics and data science is to make a scatter plot matrix, very, very easy to make in Matplotlib. But if you try to do this with genomic data, this is just one scatter plot, and it's not really informative. I actually jittered the data to increase the amount of variation. But basically, you could make any number of these, and you really wouldn't get a snapshot of what your data is. So we need to use some cooler tools in our data science toolkit to start plotting and looking at these data. And so PCA, if you're interested in data science and statistics, you should get to know PCA. It's well worth the effort, plenty of tutorials and blog posts on it. And what PCA does, it lets you take tens, hundreds, thousands, even millions of columns of data and boil them down to an arbitrarily small number. 2 or 3 is pretty common. We'll use 2. And I kind of think of this as like the genie in Aladdin. There's all this cosmic, immense data, but we can boil it down to just a tiny little itty-bitty living space. So that's what we're going to do here. We're going to take 15,000 columns of data and boil them down to just an x-axis and a y-axis. And we can end up getting a plot like this. And this is very, very common if you look at almost any data set that has global genomics information. You're going to see a plot like this with wide separation on the x-axis. And this x-axis is representing about 9 or 10% of the amount of variation that is in the data. And what this kind of means is that these folks over here, the blue folks over here, most of them, say for a given variant, a given spot, they are all AA, and maybe some of them have A and G for a particular place in their genomes. And then these folks over there on the right, most of them are AG or GG. So they all have, everyone possibly has an A or a G, but most folks have an A or two on the left, and most folks have a G or two on the right. The y-axis then represents variation among these individuals over here, and it's kind of a long gradient where, for example, most people perhaps are GG down there at the bottom, and then they're AG in the middle and AA at the top. Key thing about genomics data is there's almost nothing unique about any given group or people. So it's not like you rarely ever find a population where everyone is GG. It's just that might be the most common condition for that particular part of the genome. And so the x-axis in this data set happens to represent individuals that are from Africa versus everywhere else. And the y-axis represents all of the variation and the processes that occurred as human beings spread across the rest of the earth. So left and right is the genomic impact of us moving out of Africa 50,000 or so years ago. And then top to bottom is variation that panned out as we migrated around the rest of the globe. So these plots, sometimes you see them on some of the darker corners of the internet, and people saying, well, like, Africa is so different. Look at it. There's this big gap here. But that's not really true. It's a dramatic simplification. And what really all this is doing is showing you some subtle differences that our most powerful mathematical tools can tease out with a lot of data. And first thing is, we have to remember that this is an analysis that is using a small subset of our genome. 96% of the genomes of all of these people are almost identical. So we're looking at we're really honing in on just the parts that vary and ignoring the 96% that is the same. And actually, while they're showing visually there's this big gap between the left and the right group, there's actually, there's not that much variation there because it's only 9% of the variability in the data set is being represented by that x-axis. And there's lots of other ways I could break this down and talk about it, but kind of the biggest thing to remember is that big gap in the data set. And this is a key thing in all data science and machine learning and deep learning, and especially in genomics data where all the data is still heavily biased. This gap here would be filled in if we spent the time and money of sampling North Africa and Egypt and the Middle East and the Mediterranean more thoroughly. So this would turn into a long smear, and there's better data sets now that aren't open access, but you can see how that gap gets filled in with a better sampling regime. One thing that's cool, once we start playing around with these data, we can take our own 23andMe data or data from individuals that have it online, and we can run it through our machine learning, and we can see where we fall. So we take all of this data, we run it through our PCA, and we get an X and a Y coordinate, and we can put it on a plot. And so the individual whose data I'm using, they fall out when I run them through the algorithm. They fall out down here. So as best I can tell, they are of Central European ancestry down there. And that's just using PCA. We can now use lots of other algorithms and things here. I'm going to skip over this one. And the last thing that I want to cover here is what 23andMe, one of the main things that they do and they market is they can take your data and they can try to predict geographically where your ancestors originated from, Europe or India or China and so forth. And so for this person, we can put it through a multi-class algorithm, multi-class classification algorithm, and we can get a score for different positions that are in the data set. And the big thing is this is entirely database dependent on the data set. You can only get possible answers for the labels that were in the original data set. This data set has in Europe, Central Europe, Northern Europe, Slovenia, and Tuscany, and this person is ranked the highest as being in Central Europe. But that just means that's the most similar. The algorithm has to provide an answer, and that's the best answer, even if the person is from someplace else. So lots of other great stuff. You can do deep learning and all sorts of things. These are my take-home lessons. You can play with genomics data, you can download it, and you can do lots of cool things. So go ahead and take your picture. I'll put my last slide. But thank you very much. This is something I'm very passionate about. I'm leaving academia to do other stuff, but this is my pet project, is figuring out how to get folks like you interested in whether playing around with open genomics data would be fun or not. So, all right. Thank you very much.